Thank you for your cooperation. 尊敬的各位来宾，会议现在正式开始。Ladies and gentlemen, the session now officially starts.
Well, good afternoon. For those of you who enjoyed joining us for this morning's session, this is going to be a continuation with a broader discussion among other distinguished guests. Let me once again introduce our award winners for this year. They are Carolyn Luger, sitting just to my left, Daniela Rhodes to her left, and Tim Richmond to her left. And we very much enjoyed their lectures this morning, and that will serve as the beginning of a longer discussion that I hope to have you join along in the conversation. So let me finish around the room here. We have Dirk Gerlich, who was last year's WLA award winner, the very first one. To his left is Ulrich Hartl, a distinguished scholar from the Max Planck Institute in Martinsried, Germany. To his left is Gero Miesenbach, who was a distinguished scholar, having won the Shaw Prize in Hong Kong a couple of years ago. He's in Great Britain as a professor. And to his left is Zhu Yanwei, who is a professor here in Shanghai at Fudan University, a crystallographer, and he will certainly want to join in the conversation with our experts. To my right is Michael Levitt, who was a Nobel laureate in 2013, so we shared the stage that year in Stockholm. He is a professor at Stanford, or has been a professor at Stanford, still are a professor at Stanford. To his left is Cao Jiafeng, who is a member of the Chinese Academy of Sciences, and as she told me earlier today, she's a colleague of my partner's Professor Sabia Merchant at UC Berkeley. And to her right is Susan Gasser, whom I introduced this morning. She is the director of ISREC in Lausanne, and before that she was the director of the Friedrich Miescher Institute in Basel, and it's been my pleasure to have her serve on this committee for the past couple of years, and she's taken a leadership role this year in particular, and so I'm so grateful to her for that. Well, I want to kick it off by starting with some questions to our award winners this year. We saw this morning the power of structural biology to solve an enormously complex structure and whose features have only since then really taken shape with an understanding of the regulation of the process of nucleosome sliding and movement and control often by post-translational modifications. So what they did was an enormous technical achievement. You saw the difficulty of that. What I want to ask each of the laureates this year is what roadblocks did they encounter, and how did they overcome this? Because this is often the question that young people ask me. How do you maintain an interest in a tough problem in the face of the constant failure that you must somehow encounter and embrace as an experimentalist? So, Carolyn, I'll let you start with that.
Well, I don't. I think it's. I don't like to give up, and so once I've sunk my teeth into something, I just continued with it. But it was very hard, and I think as a scientist, what I tell my students is you have to learn from experiments that don't turn out the way you think they turn out, and so you get a result, and it's a failed experiment, and the gut instinct is, oh, I was just too stupid, or I forgot a buffer, or I did something wrong. But eventually you have to listen to the failed experiment, and maybe it's telling you something and it takes you into a different direction. And that was definitely the case for one of the key experiments that I had to accomplish to generate uniformly positioned nucleosomes. And you can look at the iBio talk that I flashed up earlier during my talk to give you the gory details. But it's really listening to experiments that did not work. What can they tell us? And how can you learn from them? And how can you move on from that? That was really the key in this whole endeavor, and then going forward as a group leader and PI.
So I agree with everything that Carolyn already said. I think, though, I want to step back one bit more. I think, particularly when you're young, it's really important, well, I think always important, that you work on something that you're passionately interested in. So that believe that the problem you're working on is the most important problem. And I think when you're young, it takes a while to realize that actually in the lab we fail most of the time. So you just have to have the stamina to think around the problem and how to tackle it from different angles and stick to it until it works. Eventually it will work. You have to believe that. So, yes. And the other is, although it's very hard that we fail, once you get the result, even a small result that indicates, as you say, what the path might be, it's extremely rewarding.
Well, those are the basic ideas, all right. The passion one is really very important. One must be very interested. The thing that I like about science a lot, and I don't know if people think of it so often, is the scientific method. The scientific method in science is basically the thing that guides me. I love making a hypothesis. You might think solving a structure is, well, we know how to do that, you turn the crank. But it's not true. One has to figure out how you're going to solve it, and you have to figure out how you're going to make these crystals better, and so on. And therefore, you have to make hypotheses about how to proceed. And of course, they're not always going to be right. But if you think of it in that framework, that's no problem, because that's the whole point of science. You follow the method, it fails, you come up with a better hypothesis, and eventually the problem will fall. And also, in terms of barriers, you have to keep your eyes open at the failures as well. I mentioned today that we used this non-volatile alcohol to improve the resolution, and I used that. So what actually happened is that because we had a little bit in there all the time, Daniela put in 5% or 2% hexane diol in the crystallization, because she knew that when she was working on the tRNA problem, transfer RNA, that it helped stabilize the crystals, so they put it in for the nucleosome. Well, we searched, as I said, hundreds of different heavy atom derivatives and concentrations, and one of them, the capillary, cracked. And we saw, the crystals are diffracting to slightly higher resolution. We didn't know the capillary cracked, but we had to go back and investigate and try and figure out. So the point is that even in your failures, you have to keep your eyes open to see what happened, and maybe it can be of use to you. And in that particular case, it made a huge difference, because I could play with it now. I knew what a variable was. I got to higher resolution, and I solved the problem of isomorphism between the crystals. Anyway, that's just an example.
Thank you. Let me raise a more specific topic of mutual interest that surely you've thought about, and I know others around the table I'd like to engage in this conversation. We've recently seen a sea change in protein structure predictions with the advent of AlphaFold, which seems to have swept over the world quite remarkably fast, so much so that I was at a Gordon conference this past summer where every talk, irrespective of whether they had a protein sequence, was somehow an AlphaFold of something or other. This was so influential that 2 years ago, it was declared the discovery of the year at Science magazine, and the editor made the unfortunate mistake of writing an editorial to say that crystallography was more or less done at this point. He, of course, was upbraided by a comment written back to him by a group of crystallographers. But let me start by asking a provocative question to the three of you. Have you ever had a look at an AlphaFold program to see if it could come close to what you've already determined in terms of the structure of the nucleosome? Can it actually tackle the structure of a ribonucleoprotein particle?
Well, there was just a recent paper showing remarkable advances in nucleic acid prediction, protein structures, and I think it is, I will say, as a defense of crystallography and also more recently cryo-EM, that AlphaFold is based on those many, many crystal structures that have been done over decades and decades of time. And by the same token, the advances in cryo-EM are only possible by the hard and painstaking labor of crystallography of protein domains and smaller structures. So we see this quite often that thesis committees will say, oh, well, solving a cryo-EM structure, is that even a thesis anymore? And that's emphatically not true. Having said that, I think looking at structures in situ through cryo-electron tomography is really the future, and that's, I think, where the field is moving to. And it has all the difficulties and all the challenges that cryo-EM had in the earlier days, and those will also be overcome, no doubt.
Well, I would have something to say about that with respect to the nucleosome. It would be a dream of mine, or it was a dream of mine, that I could predict where the histone octamer, where nucleosome, the core sat over the entire genome. I know where these positions are, I know how mobile the octamer would be, and I know how stable it would be. Now, if I want to apply AlphaFold to that problem, I think I have a problem. First of all, I don't even know if AlphaFold could predict the structure of a histone. I just don't know that because I haven't bothered to look. But my guess is it would have a problem because there aren't enough homologous sequences. It's so conserved throughout eukaryotes that there's not going to be enough data to do it. So what would it take to have an AlphaFold-like program tell me exactly where the nucleosome core is in a genome? Well, first thing, I need a database then of structures with basically many, 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 many different sequences. I'd have to know how they sit. I'd have to get the structures so I could see at the level that I showed you today about kinks and smooth bending and all that sort of thing. Unfortunately, it seems a long way off. Could I tackle, maybe with a robot, assembling nucleosome cores with different DNAs? I sure could. I actually had a grant to do that, but I couldn't find anybody who would do it, who would work on it. But one would have to use, Carolyn showed she got a 2.6 angstrom structure with the cryo-EM. I'm not sure that's good enough. I'm not even sure, when we really could see what was going on with the parameters of the DNA, what I would actually want to use as a large database, we needed 2 angstroms or better. Cryo-EM, if you're willing to take enough micrographs and spend enough time, might be able to do it. I just don't know. It's never been tried. So when it comes to the mechanistic detail, and as Aaron Klug used to like to say, the devil's in the detail. It really is. You have to know these things if you want to advance. Whether the cryo-EM will get you there eventually, whether crystallography still has a place, when you say, I want to know the mechanism of the HIV integrase exactly, I think you're still going to be doing crystal structures for a while.
But the only thing I like to add from my colleagues is, I have not used AlphaFold, I'm too old, I'm an experimentalist, but I think now mostly we are interested in multi-protein complexes. Most complexes contain tens of proteins. And although, from what I've read, the AlphaFold is able to with some surety have the local fold of proteins, I'm not convinced yet that you can determine the interactions between, I mean, the really interesting thing, how a complex is assembled from individual proteins. And I know people are working on interaction surfaces, but they are very complex, and I don't know, maybe Michael would know better than me, surely would know better than me, and in the end, as an experimentalist, yes, I can do the prediction, but at the end I would want to see the structure, you know, and nowadays by cryo-EM, obviously. Okay.
Okay, well, let's turn it over to Michael. You're our local computational expert. How far more?
So I think we need to understand that AlphaFold, using the word fold here, is a big misnomer. It's basically AlphaPredict, if you like, or alpha homology model. We've just saturated the sequence and structure space so well that an awful lot of interesting proteins, say for drug design, are relatively well predicted. But again, when the folding problem was first, I mean, a protein can fold its single sequence. It doesn't have to ask all the other variants. AlphaFold depends completely on correlated mutations that are occurring in a family of sequences. And if you have these either with inner chain or between chains, you can do it. It's in no way, and again, when you use AlphaFold, for example, there's no way you can actually ask the question, could AlphaFold predict the nucleosomes? Because they're already in the database. So, you know, so in some senses, it's a very powerful database. I think that if you're going to work on a new structure, check it, make sure, you know. And the other thing is also, for some tasks, people are saying that the sidechain detail is not high enough. And I don't know. You might also argue that if you want to do drug design, often you want to be able to co-crystallize compounds with it. On the other hand, people have actually used AlphaFold structures to get new drugs. So I think it's a really important advance. But I don't think in any way it negates what people are doing. I think you would be making a mistake if you ignored it in doing something new. But I believe that it just provides a higher starting point.
Let me ask now, Zhu Yanwei, you're another crystallographer on our committee here, on our so-called roundtable. What do you have to say about the application of AlphaFold as opposed to experimental derivation of structures?
I completely agree with the three Laureates and Michael's comments. For example, in my study, I studied the transcription initiation complexes consisting of 84 polypeptides with a molecular weight of about over 4 megadalton. So when I change the several DNA sequences, the whole complex will adopt quite distinct conformation, very large different conformational changes. So I thought that AlphaFold may get such prediction for the complicated processes. Also, for the detailed, as Tim mentioned, if you want to make a drug, a small molecule interaction with the protein target, it will not get enough information to guide your design, the compound. So, so that's the current limitation for AlphaFold. But in another way, we think this tremendous advances, it does help us a lot. For structural biologists, sometimes we obtain low resolution maps, so we can dock the predict structure model into the map, to help us to build a more accurate model. So that's helps a lot. In other research areas, other researchers may not require such precise information. Just get an overall fold for their interested proteins. It also helps a lot to get their mutational functional studies. Yeah, that's my understanding.
Another area where AlphaFold has made no progress, and even traditional methods of structure determination are challenged, is in your area of research, Dirk. These hydrogels and liquid-liquid phase separated organelles. How are we going to solve how those structures form of relevance to, for instance, what you've discovered on their role in nuclear transport?
Yeah, so that's another limitation of AlphaFold. But let me answer, or reply to your first question. AlphaFold is super powerful, but one always should be aware of the limitations. So we had a really nice experience. So we made nanobodies against SARS-CoV-2. And soft crystal structures all look great. And then we tried to predict the effect of escape mutations. Just a single mutation, Q493R, one of the Omicron mutations. AlphaFold built a beautiful model. Yeah? So the arginine in hydrogen bonds of ideal geometry. And when we measured it, the affinity dropped from 10 picomole to not detectable. Yeah? So AlphaFold just recapitulates what it has learned. And if it's encountering something it has not learned, it fails. It may fail badly. Yeah? Or another example, it is absolutely terrific in predicting structures that are homologous to what it has learned before. We look at nanobodies. So these are small antibody fragments, 15 kilodalton, 130 amino acids. Looks very simple. But the immune system invents new ways of fold. Yeah? So, and even just predicting the fold of a long CDR3 loop, AlphaFold failed quite badly. Yeah? So this doesn't say it's a bad method. It's just to say that you have to be aware of the limitation. And well, the other limitation is intrinsically disordered proteins. So it makes up maybe 1/3 of a mammalian proteome. And it's just resistant to this kind of prediction because they're flexible, they're not a unique conformation. And if you ask me how to predict their structures, I would love the answer. The problem is when these intrinsically disordered domains interact with each other, it's just a space that has so many dimensions that I don't think any computational method at present is able to predict this in a reasonable way. So I'm hoping for an AlphaFold revolution for intrinsically disordered interactions.
Okay, don't lose any sleep over that one. I'd like to change the subject just slightly to engage most of the audience, and that is young Chinese scholars. And perhaps I can ask Gao Zhangfeng how you feel this organization, the WLA, can contribute to the advancement of science, of basic science here in China.
I think it's very important for basic science. Currently, many of the fundings assume they try to get more people working on more applied science, meaning it's important. Actually, in my point of view, I think basic science, deliver more than what we really see. So people, for example, very, very basic DNA double helix, long time ago, 70 years ago probably. And people don't know by the time what is going to be useful. But now everyone use genetics code to, do all this medicine and agriculture type of study. So to me, I think this meeting is very useful to let all these young students or postdoc or young scientists to know how important for the basic science and learn from all these well-established people. and get all this spirit experience, seeing, like Carla said, not so easy, it's tough. So that's to everyone, not only for you young people, to all these well-established persons. So I think it's very important.
So the question here in China is, who are the people in the decision-making power that decide how the funding is allocated? My impression from a distance, knowing that I have several former postdocs who are back in China, actually the support for basic science, from my perspective, seems pretty good.
I think to some extent it's pretty good, but in the long-term run, I think some big money is still quite focused on all this applied part. Yeah, and we are getting good to get some private funding, which are more towards to basic science. Yeah.
One of the things that we can do here is by promoting the great discoveries that we recognized last year and this year to demonstrate how basic science does have application. It surely will, and there are so many examples where it has. I think our intention with this prize is to continue to recognize basic science.
Yes, exactly.
Maybe I can engage the other members of the committee, all of whom have either served on prize committees or won prizes. What good do these prizes do? Is there any value other than to the individual in having these prizes? Susan, you're on the current committee. How do you answer that question?
Okay, yes, okay. Well, it's not for the money, if you want to know the truth. It's for the recognition. I think what prizes do is put the into perspective, actually, how we all build on the shoulders of others. We have 3 today, but I could have named 10 others who contributed to this prize winning, and it's quite difficult actually to sort out who did what and which was the most insightful piece in the puzzle. But I think what it does do is inspire you to realize that, 40 or 50 years ago, or 30 years ago even, we didn't really know, how do you get rid of a nucleosome to allow transcription? How do you manipulate this? What are the charges we have to neutralize? There were so many unknowns. And so what I think a prize can do is inspire people to actually dig deep, but at the same time keep the big question in mind. Remember that science has a tendency to make you narrower and narrower. And we have to do that. We have to focus on the details. But in the end, the impact can be very broad. And so you also have to think that helps guide you through the maze of details. So I think prizes highlight what those big questions were, even if at the time we weren't really sure. We knew it was a big question, the nucleosome structure. That's why they were passionate about it. But it wasn't really clear what it would tell us. And I think, it keeps you going in times of detail. Prizes highlight the big question.
I think another important element of prizes is that it's an occasion where the public, the non-scientific public, learns about science, learns that there are scientists who do this type of work and in fact do it with the taxpayers' money. And this is certainly true for the most well-known prizes like the Nobel Prize, where on this day everybody in the world learns a little bit about science and about this particular discovery that is being recognized. And I think it also is then an inspiration for young people perhaps to go into a scientific profession, to study natural sciences. And in that regard, I think it has a very important purpose beyond recognizing the specific scientists for their discoveries.
You have experience in this yourself, so how do you feel about it?
Modest experience, yes. I think prizes are a double-edged sword. I mean, on the one hand, they are some of the greatest PR that the general public sees about science. I think they also highlight the importance of imagination, that a discovery is not just a piece of data, but it's usually the conceptualization of that piece of data. And the really great discoveries are always based on a model that the discoverer has of how the hidden processes that give rise to what we can see and measure actually operate. This is where the real insight comes from. When I say prizes can be a double-edged sword, I mean that they are often a little bit anachronistic in terms of the limitations that they impose, because as Susan has said, there's often many contributors to a specific advance, and it's often difficult and sometimes cruel to draw a line.
The Nobel Peace Prize occasionally will go to big groups. Well, maybe we should do that for the basic science awards too, somehow. Someone would always be left out, feel cheated somehow. Okay, I want to...
Can I
just
add something here? I think following on from what both Hartel and Gero said, I think that a very important aspect of prizes is that in some senses you are singled out, and I think, although it's never stated, you're expected to involve yourself in outreach and basically be more exposed than you perhaps would be otherwise. I have a very distinct memory of when I heard about the prize thinking, what's all this nonsense? I'm exactly the same as I was 3 hours ago. And then I remembered the awe that people like Crick and Klug and Perutz, I mean, all the people at MRC where you had 15 Nobel Prize winners had on me. I was interested in what cars they had, what girlfriends they had or didn't have. I mean, because it was basically seeing scientists as real people. And I think it's this that is actually very, very important. Obviously, it's not the selection criterion. But I think it's part of what, you know, say for the Nobel Prize, the way that they stop the traffic in Stockholm. I mean, they make a point of being ridiculous at the level of sort of how important it is. And I think this is done partially to sort of make you realize that, you know, you're going to have to do this silly stuff for a long time ahead. And I think it is important because basically nothing is more important than encouraging young people. Something that worries me very much is the aging of the world. And very soon the median age will be over 65. And that is really scary. So, because young people are still young people. And I think we have to go out of our way to encourage them to be original and think. And, you know, I think a real sin of science has been to allow these fancy journals to keep people doing postdocs until they're 40, which is at a time they should have been totally independent for 20 years. And 40 is not the new 20. There is no new 20. 20 is the 20. And I think there are issues like this that prizes help you emphasize.
Yes, sure, Tim, please.
So is this on? One Nobel Prize winner that I knew quite well, Aaron Klug, I can tell you what it meant to him. He was asked, what would he do with all that money? He said, well, you know, I have to go between Cambridge and the lab twice a day. I need a new bicycle. That was it. I mean, it meant a lot more than that, I'm sure, but I just thought that was a very curious thing for him to say.
He could have bought a lot more than one bicycle.
Actually, he didn't have to spend any of his prize money on it. They just started appearing at his door.
I think one little detail useful about prizes. So my granddaughter who's drawing a show, I learned that in Sweden, in infant school, they celebrate the Nobel Prize. The children have a Nobel party. And I think that's a very nice way, actually, of exposing the children to science. And I have been asked to go to the infant school to talk because I won this prize. So that's a very nice invitation.
I'd like to pose a question to each of you, and we'll go around the table for your answers. So I don't know about you, but I'm often asked to look into a crystal ball and tell the audience what's the next great thing to be done. And of course, I have no answer to that. So I'm going to ask all of you for an answer to the following question. If you were in your late 20s and able to start your career all over again, which question would you pursue? So let me start on the end with Zhuan.
That's a question that I really want to ask the laureates. So to me, I'm interested in the the extension of my current study, the transcription. So we want to understand how transcription occurs on the complex quarantine environment. So that's really the very challenging question that cannot be fully addressed by current technology. For example, the cryo-EM, the imaging, the sequencing cannot fully address, tell us how that complicated process occurs on the polynucleosome. For example, in cells, it's highly dynamic. So I think the advance in technology is required for fully understand this question. That's what I think in the molecular biology in the future, there's a question need to be addressed.
So I'm a neuroscientist, and I think in my area, the big unknown is the human brain. And within the next few decades, there will be methods to study the human brain at the resolution that matters, namely individual cells, individual electrical events. And they, if they succeed, will really transform our understanding of ourselves.
I guess most of us would probably say they want to become a neuroscientist if they had to start again. But I think something that's a bit closer to what we actually work on, it would be fantastic if we could take a cell apart and put it back together again, even the simplest form. And I think we are very, very far from that. And the future will have abilities to build cells and help us in understanding fundamental principles. We don't understand yet what is really the difference between our in vitro experiments that we all do and what happens in the cellular environment, what characterizes it. I'm now working on ways to observe the molecules that we are interested in their natural environment, to see how long they interact with a particular substrate, and how this is different from the in vitro situation. Obviously, these reactions are so robust that they still work in the test tube, but that's far from the conditions in an intact cell.
Well, that's a difficult question. I mean, in the end, you know, it's sort of predicting the outcome of science, and you can take two perspectives. So there had been science projects where the question was clear from the beginning and took decades to solve it. I mean, this is what we heard today as an example. There are other discoveries that came out of the blue. No one had an idea that they exist. I mean, think of restriction enzymes, think of RNA interference, think of CRISPR. You know, the people who discovered this never had the plan to do it. And, you know, it was not, I'm not saying it was not planful, but it was an explorative way of doing science, a playful way of doing science. So this is quite close to me. And, you know, considering all the knowledge today, you know, I would wonder there is something totally unexpected. I mean, people think of life on Mars or in other solar systems. I actually keep thinking of exotic living matter that is understudied, and where one might find different solutions to common biological problems. I mean, it's like mathematics, yeah? So a good mathematician is not just happy by solving the problem, but finding all the solutions. And in terms of life, I think it's interesting to also get a taste of how many different solutions would be there. And if I would start again, I think I would start something totally exotic, underexplored, complex organism, ciliates, for example, which have 2 nuclei that import different things, have type of vacuoles that don't exist in any. I would go for the exots.
Good luck. I was hoping you wouldn't come to me. That's a really tough question. But actually, I haven't really stopped doing things. It's just harder to do what I had done before without a lab. What I can do now, and probably would start with earlier on, is try to understand computing methods well enough to make new applications. So in the last few years, I've attempted to understand quantum computing. I think I can understand neural network computing. And the computing aspect is great, but I don't really see that as where I'm going. The nice thing about the neural side is it ties you into the neurobiology. So any advance in neurobiology, you can think, well, how could I use that in the computing? But then it has to have an application. And the application is the same application that I've been interested for a long time, and that is, how does gene transcription work in eukaryotes? So to take that kind of computational approach to the new developments that come out in structure and otherwise, following more or less a path of Michael Levitt, carrying that on to try and actually understand these things better using computational methods. That's basically what I would do.
If I were young again, which is hard to think about, I think, like many of us, I would want to be a neurobiologist, because I think understanding the human brain is a real challenge. But closer, I think more obvious, I have said for years, if I had more years to work, I would definitely take a much more global look at nuclear architecture. How the nucleus is actually organized, rather than what we have done so far, looking at small details of what happens inside the nucleus. I even look at things, going in and out of the nucleus and how they talk to each other. I think regulation of nuclear architecture would probably be what I would be working on.
I'm actually with Dirk. I'm all for looking at the weird things and the unexpected things. And the analogy I'm using a lot is the gold rush. People who knew there was a gold rush and there was gold to be found, went there, never got rich. It's the people who just wandered about the Arctic tundra, stumbled on a gold mine. Those are the people that got rich. And so I think following up on the weird is really a powerful approach to fall on your face 1 time, 2 times, but maybe sometimes you discover CRISPR or you discover restriction enzyme or something really powerful. Now that is of course very risky, especially if you're young and you're trying to make a career. But I think having funding from private funding organizations that maybe see the long view and let you fail occasionally is really powerful in that extent, because that really has the power to make quantum leap discovery. Now, having said that, probably a more mundane goal, I don't want to be a neurobiologist. I actually really don't. But I just want to watch a cell going about its business at atomic resolution in time-resolved manner, just watching it do its thing, and then maybe building a mini organelle and watching it talk to its fellow cells. I think that would just be amazingly cool. I like to figure out how stuff works, and that would really be very satisfying and very easy to achieve, I'm sure. Really easy.
Well, the part about the question that I posed that I like the best is the idea of going back to being 27 again when I was the beginning assistant professor. That would be the best part. But I'm going to answer the question by not picking a particular area that I think would be ripe for attack now, but rather how I would recommend that young people think about their career, looking prospectively rather than retrospectively. When I was a graduate student in the early 1970s, I worked with a remarkable biochemist by the name of Arthur Kornberg, Roger Kornberg's father, and he was perhaps the best biochemist of his generation. So I learned a craft from him of how to take a complicated problem apart piece by piece. That field at the time, DNA replication, was already so competitive that I found it really uncomfortable being in that area or even thinking about what I might do using my experience in his lab to continue. So I resolved, as a result of that discomfort, to find an area that was underexplored. And I chose the area at the time of biological membranes. That's not the one that I would necessarily favor now. If I were to start over again, I would probably pick a different field. But my decision would be guided by the idea that there is so much to biology that is underexplored and indeed unexplored. And yet I find that too often graduate students and postdoctoral fellows are so conservative that they just decide to, you know, they've done well, they've published their Cell or Nature paper and they're on a roll, and they just continue to turn the crank and do more of the same. And I think that, unfortunately, that behavior is reinforced by the so-called prestige journals, because these are the hot topics that get published in those journals. But I think that is not necessarily the way progress is made. So I would, for the young people who are faced with this opportunity, I would urge you to think differently, or as Steve Jobs said, think different. Do something different. Don't just keep turning the crank. Look for areas that are underdeveloped and be your own master.
So the question of what one should do next is a perennial one. And my normal answer is, whatever you like, we have no idea. You might as well do something you like doing. Because I do think that the future is intrinsically unknowable. And anyone who thinks that, for example, we should stop working on bacteria and only work on eukaryote cells is just making a fool of themselves. So I think you have to be very careful about recommending. But I do think that we're at a very special time now. And my guess is that none of you have been playing with large language models like I have. But I can say conservatively that GPT-4 is to our whole life what AlphaFold is to protein structure. It is a radical change. It's hard to even estimate how big the change is. I have probably asked GPT 5,000 questions in the last 11 months. I use it on my phone, I use it on my computer, I use it for everything. And as a summary, I would say that it's made me 3 times more productive and 25% smarter. And given that it's a bell-shaped curve, 25% really gets you into the rare phenomena. So I would say right now, everybody needs to do in exactly the same way. When the phone came out, a few people said, oh, I don't need a smartphone, I don't need a phone at all. Who wants to make conversations? If you basically cut yourself off from the internet, smartphones and computers, you'll go back, your IQ will drop very, very significantly. GPT is a radical, complete change. It's a total game changer for anything, whether it's psychological problems, family problems, Python programming, understanding electronics. It is helpful in every one of these things, and probably 500 more things that I haven't tried. It translates languages flawlessly. If you all wanted to speak loudly in your own language, I would get a translation within a few seconds on my phone. So these are amazingly powerful things. So I would say right now, get into the modern age and then decide what you want to do.
So I Actually, my background was biochemistry when I did my undergrads, and I did postdoc with Steve Jacobson, probably you know, starting on epigenetics, basically working on DNA methylation. Since I came back, I work on more on histone modification. That's why I read quite a lot of paper from Carlos, and your slides was always on my lectures, you know, to teach the other graduate students. So I think if I start one more time, once again, in my, you know, I would like to do more towards to more broad rather than biochemistry, biochemical level study, especially I work on plants. Because plants has to deal with all kind of harsh condition. And plants, we want to have a high yield tolerance to all kind of stress condition. I would like to learn more from plants and the interaction between the plant and the soil. So after the plants have been growing for so many years, the soil quality drops greatly. So there's some, you know, interaction between plants and the soil. So I think that will be a very broad area. And in old China, you know, it's a very traditional agriculture types of society. The soil used to from the farmers, the nurse of the soil make their family's soil richer and richer. But for the modern ones are, you know, use the chemical fertilizer, the soil quality drops greatly. So there's something between we need to take good care of it. And there's many basic questions. For example, all these wild species can grow nicely like, for example, for the grass, they can grow into very poor quality of the soil. So basically, they have very strong stem cell. What's the molecular basics on those? Something I would say more broad, try to understand why the plants are so strong to survive in all these tough conditions, and eventually can help human being for a sustainable agriculture or for green agriculture. That's my,
Okay, the last bit of wisdom here, questionable. But first of all, I think I'm fascinated by neurobiology also. The human brain is just so complex, and the whole idea that we have a spirit and in our brain anyway, somehow concocted of a notion of self. But out of that, I think it's very important to think about life sciences in contrast to physical sciences. And kind of the whole notion of life is sort of anti-entropy, because the whole fact that we live and grow and organize things against, you know, the drive of physical forces, which would be toward entropy, is fascinating. And I think we try to describe life, I mean, living phenomena, cellular phenomena, developmental biology in terms of physics, but in fact they fail in many cases. And I think the creative spirit of the future will be to try to find new ways to define the rules of living organisms that may not actually simply, they build upon, but they go beyond the physical laws that we mostly use to define what we know, like structure. So there's a lot more out there to discover.
Okay, well, what I'd like to discover now is if you in the audience have questions so that we can have you participate in this conversation. So I'm going to walk right down the aisle now and ask for those of you who have questions to stand up, identify yourselves, and ask our panel of experts. Questions? Yes. There we go.
Hello. Thank you very much for the discussion.
If you could identify yourself.
Yeah, my name is Eneko Larrañeta. I'm senior lecturer in Queen's University in Belfast. I work in pharmaceutical sciences. So whenever you discuss about the curiosity and the, to look, I really like the analogy of the gold rush. And what advice will you give to young researchers? Because now just the system is quite focused in producing outputs, and that's a little bit against, like, the curiosity, and sometimes it's conflicting. What advice will you give to young researchers and young scientists on that topic? Thank you very much.
Okay, I saw Daniela go for the microphone first.
I think we need to work hard on changing how science is funded, and work on politicians, actually, on how science is funded.
Yeah,
and how it is published.
And how it's
Yes, I think it's taking a really good direction with bioarchives and people publishing preprints. There's really a paradigm shift now in the publication industry where the power, shall we say, is of the big journals is getting a little more diminished, which I think is a good thing, because these journals often have way too much power over the careers of young scientists. And I think we also have to generate a culture where failure has to be embraced, because it is simply part of the process. If you know that something works, it's almost not worth doing. And the harder something is, the more likely it is going to fail. And we have to put a more positive spin on that, I think.
Thank you. Other questions? Yes.
Thanks a lot. Now we. I'm Ding Xiaohan from Shanghai Jiaotong University. I'm an undergraduate student in the third year. Now we know a new area is called bioinformatics, and I'm very interested in this area. But I have a question that I would like to ask, that how can we connect bioinformatics with our experiments better, so that we can use these new tools to prove our experiments, so that we can understand the basic biology better? I just found that there may be some gaps between these new technology or tools with our experiments. Yes, that's all. Thank you.
Can I answer that? I mean, the others may come on, but genetics. Genetics is the proof. I mean, modulation of what you predict based on bioinformatics, you have to test it in vivo with genetics. To me, there's still nothing. You can model things based on bioinformatics. Yes. In the end, you got to put it in the organism and see if it does what you predict. And I mean, in the long run, it might be creating a cell or creating something, but genetics is the way.
Okay. It's like AlphaFold. Its prediction sometimes doesn't meet our requirement, yes.
I think you also have to expand, because genetics is the answer, but you have to expand the number of model organisms that we can study, because right now we're still very limited in the types of organisms that we can genetically manipulate easily. And I think with CRISPR, that's changing rapidly.
Oh, thank you. Thank you very much.
Thanks for the question. Yes. I'll come with the microphone. Please identify yourself first.
Thank you so much. My name is Zhao Binghong, and from East China University of Science and Technology. Actually, I major in energy science, but I'm really interested about life science and medicine, because I think they both really meaningful to all the people. But actually, as a university student, I wonder how can we apply knowledge into practice, or maybe because I'm in the third grade and not really can apply knowledge into practice. So could you give me some advices? Thank you.
So you're asking how to translate basic knowledge into general practice, for example, for medicine? Was that the question? So I think, was that the question? Sorry. Yes? She's still.
I wonder how to apply knowledge maybe to practice.
Well, I mean, if you talk about translational aspects of research, it's really going out and talking to industry leaders and maybe talking to startup companies, seeing how you can translate what you've learned in basic science to applied sciences. I'm not sure that answered the question.
Actually, one thing is to understand what are the big questions. For instance, medical questions are often, doctors understand them, maybe patients understand them, but scientists rarely do because we live in another world. So it's very important to interact, to do this interdisciplinary contact between the technical specialty that you may master and the source of the real problem. So at least for medical problems, it's very important that scientists today actually learn to talk with doctors and patients to understand what is the problem, where we can apply all of our knowledge to solve, to make progress. So often just understanding what the problem is halfway.
Maybe I can just add, in my career, I've seen an enormous change in the connection between what we do in basic science laboratories and what gets done in biotechnology and drug discovery and in the pharmaceutical industry. When I was a graduate student, no one went to companies. Everyone was a pure academic doing just basic science. But that changed completely. Many of our students now go into biotechnology. So that transfer of knowledge from the laboratory to technology to the clinic is much better, much easier than it used to be.
Hi, thank you. Renata Jurkowska, I'm from Cardiff University. I'm also kind of one of those who just started their own independent career. But now that Susan mentioned, kind of a question popped to my head as well. What can we do, or what can the most prominent do to push on a society level that we bring those sectors together? Because you mentioned justfully, I came actually the other way around. I came from company to academia back. But it's very rare. We still have these worlds not talking to each other, and you mentioned clinicians and academics. But I would push it even more. If you want to do translational science, then how do you bring and talk to industry? And then, how do you enable these, I think we speak different languages still. So my question would be what advice or how can we promote this collaboration much better on a more structural level?
I can provide a simple answer to that. One technique that I've seen at my institution is some fellowships that graduate students are offered require that they spend a 3-month period working in a biotech company as part of their training. And the students embrace this because many of them actually want to go into biotechnology. So that eases that transition.
There's another possibility as well that scientists, group leaders, and so on organize meetings and then ask specifically to have industry representatives come. Mm-hmm. And it works generally. Mm-hmm. And you may come up with some new ideas there that can be translated. It may be even that you make such a good connection that they will then provide money for 1 or 2 or more postdoctoral students. And I've had that sort of success with things in my own career, but it takes some effort to organize regional meeting, for example, and bring in the industry. In Switzerland, of course, there's all the pharma industry, so perhaps it's a little bit easier, but still, that's another approach to take.
In the back, I'll get to you next, but first let me go back here. You have a microphone. Carry on. Identify yourself.
Thank you. I'm Jackie Maben. I'm a clinician scientist from the University of Edinburgh, so really good to hear that you find that important. I wondered if I could ask a rather personal question of the panelists, just because we have the opportunity. We've heard a lot about failure and how you learn more from failure than success sometimes. And I wondered if anybody would be comfortable sharing their biggest regrets in their career.
Oh, come on. You've all made perfect decisions in your life and never had any regrets?
I think that, firstly, everyone here has succeeded, and therefore you remember the good stuff. But we also see many, many cases where success is largely a matter of luck. I can see 20 junctions in my life where I wanted to do A and somebody made me do B, or vice versa. So it's not exactly like it's in your hands. But I think that persistence, passion, the standard things can really get you over that. What interests me is, everyone is taught to succeed. You try to get the highest mark on the exam. You try to get the best PhD. How would you train people for failure? Now, failure is an interesting thing, because let's imagine somebody said to all of us, you can never fail. You'll be funded, but just don't fail. It would be very easy. We would do really easy things. Yeah. So in some ways, if you want people not to fail, you're basically saying, don't do something interesting. By corollary, if you do something interesting, you're going to fail. The question is how do you, so you could actually argue the following. It's an exam, you answer the wrong question, but it's a cool answer. You should get full marks. We don't do this. But I don't think it's just true of science. You could argue that a bureaucrat who never fails isn't trying hard enough. He's basically sitting so far in the red line. So I think a government that never fails isn't trying hard enough. This is a this scientific method applied broadly in life says that success comes from failure. Imagine you said to a baby trying to walk, just fall down once and you're done for. No one would learn to walk. So I think we need to really appreciate very deeply how important failure is. But I think also we need to learn not to be too bothered by failure. And one of the things that worries me is that people who are, get to the best university because they get a perfect grade and so on, aren't being trained about failure. And I worry about that. They become brittle. Resilience is a very important characteristic. So, I think it's a big, big problem. Your first question.
We have a bunch of answers here. Carolyn was first.
Yeah, so thank you for that question, which is indeed quite personal, but a good one. And I think my regret is just giving into the relentlessness of the minutiae and all the things we have to do. Susan knows very well, and everybody here, committee work and this and that and the other, that really fragments your time and really leaves the time that we have for thinking hard about particular projects quite limited. And so that's my regret, and my advice would be to learn how to say no to some things and really think hard about what commitments you take on in addition to your main commitment of doing science and leading a lab and mentoring students.
I must say I don't really have any regrets, but I would like to comment on this idea of you should get used to failure. Yes, but actually it should bother you to fail. And I already sort of covered that by saying it should drive you. It should make you think harder about why you failed and how to do it better the next time. One of the things that disturbed me the most about some of the students that I had is they wouldn't tell me when they failed. They would disappear, or they would just be quiet. And that's no way to do science. You can't do science that way. You have to be completely open. And that's why, as a young person, when you're looking to go somewhere, to a lab, first of all, you have to decide on the area that you want. You can always change it later if it doesn't work out. But you need to find a supervisor who is open without saying, oh, you stupid person, you failed on that. You need to find somebody open that realizes this is a problem or these are things, and to help you go to the next step. But the idea that you should just accept failure, no, it should burn in you. It should make you dream at night about how you're going to fix it, how you're going to do it better the next time.
I'm not sure I have regrets, but I think maybe we should mention to try and get the balance between your working life and personal life. I think if you are the passionate scientist, that's a hard one. And if I have a regret, I would say maybe I didn't spend enough time with my son when he was small, because I did my PhD after he was born, okay? So do I have regrets? But at the time, it seemed to be the right thing to do. But I think many of us probably, I mean, being in science means working every day of the week and sometimes 12-hour days. And that's maybe something to consider.
If I could just add a little bit to that.
Dirk had his hand up.
I mean, you alluded to two things that are related but not quite the same. So regret and failure, there is a subtle difference. So failure, if one is thinking about science, this can be seen positively as part of a learning process. Yeah, and I should say, we learned more from the failed experiments because, I mean, a failed experiment properly executed, so with all the controls, so it's informative, you learn more than from an experiment that just meets the expectations. So the experimental failure is something very, very positive. Regret, this is more an issue of personal attitude. Yeah? Looking back, maybe should I have made a different decision? So I never look back with this kind of attitude. Yeah? So in German we have a saying, one never knows what it's good for. Yeah? And you know, even if a single decision appears not be perfect from today's perspective, it determined all your other paths, and maybe all the other positive things you have experienced since then you would not have experienced if you had made always the right decisions. So I think one should see their personal life in a positive way. Don't look back, don't regret too many things, just look forward. And it helps as a scientist, and it probably also helps, you know, in other professions.
Okay, did we help you with that one?
Yeah, wonderful, okay. Thank you.
Okay, here was a question here.
Greetings to all the professors, and I'm Jian Xiangwei from East China University of Science and Technology. And in our previous conference, I remember Professor Richmond told us that he is fond of making hypotheses. And in my opinion, there are meaningful hypotheses and there are ridiculous hypotheses. I think maybe meaningful hypotheses can lead us to successful, or otherwise you guys are not sitting here, right? And my question is, during your research career, what makes you to make a more meaningful hypothesis instead of making some hypothesis more likely to fail?
I think that's a fairly obvious answer, is that given a particular problem, you need to try and understand everything that's known about it already. And that means studying it, study the literature, make up questions in your own mind, see if you can answer them from the literature, develop that sort of strategy, and then I think your hypothesis will be good. Be reasonable, and it has to be testable, of course, otherwise it's not useful. In my own, outside of nucleosomes and life science, I like mathematics and computing and so on. And there, I don't know nearly enough. And so I make crazy hypotheses with the hope that maybe, who knows, one of them will show up. But it doesn't hurt my career to do that. I just have fun with it. But if you're in your career-wise, you really need to know what's been done and then go from there.
It also helps to discuss your hypotheses with experts in the field. So, you know, you may confront something new and you may think, oh, I've got a hypothesis that's completely new. And you should always discuss. So scientists, you know, that's why we write grants and we get reviews on our grants. Grants are basically hypotheses. And then you get comments, you know, like that's a stupid one, or that's a good one, you know. And I think and if you don't go so far as to write your every hypothesis into a grant, just discuss it. Discuss it with experts or people you respect and see what they think. Science is a very social activity.
Well, not every good science starts with a hypothesis you want to test. Yeah? So if you're at the stage that you want to test a hypothesis, you already know a lot about it. And I don't think this is how much of the transformative science is working. The transformative science is exploratory, and you just don't know what the outcome is. I mean, look at Randy, being famous for a super genetic screen to find essentially all components important in the secretory pathway. Perhaps the hypothesis was that the screen is going to work, but Randy, I don't think you had any hypothesis about the outcome. Yeah, this is just a different concept. And I should also say, if you're musing about working on a particular problem, of course you can read everything. But if the field is stuck in a state, you know, where the mainstream thinking doesn't really hit the nail on the head, if you read too much, then, you know, this will also impede your creativity. I think one should also have not too much respect for everything that is written in the textbooks and in the papers. You should always consider what you read might be wrong. Yeah? And it's just a slightly different approach. And my suggestion, I think most of the transformative research didn't follow a strict hypothesis and followed the mainstream thinking at the beginning.
Thank you.
I would just add to what Susan said is I wouldn't necessarily trust what experts say all the time. If I had trusted what experts say all the time, I would never have gotten an NIH grant. They already had rejected my views to begin with. So, question, yes.
Okay, my name is Cheng Lian from Shanghai Tech University, and my question, it's a great honor to be here to ask you experts questions. Like, I'm a teenager, so I also, for curiosity, love studying human brain, though it was not my major. But as a human brain or life, they are complex. There are very complex systems. They are not like math or computing. So when we are studying complex systems or intricate systems, they sometimes emerge some other properties. So how could we study these very complex systems, and are these jobs not, our human brain is not good at processing this mass data, and should we just take these jobs to AI or artificial intelligence like GPT? For them, they are good at computing to solve these questions. And yes.
I mean, if you look at the history of molecular biology, the way we have tackled complex problems is by taking them apart and studying the parts of the complex system. Whether we now are at the stage where we can study the whole thing, I don't quite know. I don't know. Again, Michael probably know much better than me. But I think a good starting point is to tackle parts of it and then, or different parts of it, and then put it together.
I think the issue of complex systems with emergent properties is a very hard one. I think that certainly science has always processed by taking things apart, but there's one very clear failure, and that is the properties of water. Simple liquid, but all the attempts to find a simple model for the properties of water fail. But if you put something that looks like a V into a box and shake it around, it basically gives you water. So it's an emergent property. What we care about water are emergent properties. And, 50 years ago, we couldn't do it in the computer. We didn't know the answer. Now we can do it. We still don't know the answer. So I think it's if you say to somebody, well, explain to me in the elevator why water has a density maximum at 4 degrees. It's all hand-waving. The computer can do it. Experiments can do it. So I don't know whether we'll ever, I think there are certain things that are very, very difficult. Explaining is kind of a protocol. When you explain something, you use logical constructs and things like that. And I think that sometimes we just don't have the language yet for explaining these things. I don't know, but I do think it's going to be much more common than we realize. It's going to be true, say, for the way societies behave. Societies are just a bunch of people, but they can behave in very complicated ways that are unpredictable. So, I think it's a very interesting problem, and I think we may need to define new kinds of science to deal with them.
Okay, good. Lots of questions, good.
Thank you. Oh, thank you, everyone. I'm Lindsay from Beijing. I'm working on a startup that tries to bring evidence from bench to bed a little quicker. So we're still a startup working on it. My question's a little unrelated. If I may be pardoned, just inspired by Susan's comment on a grant could be good or stupid, so this might be a stupid question, but I want to throw it out. So given the recent advancement in quantum physics, where part of my rudimentary understanding of it, where things could be really uncertain, something could be a particle or a wave, things that are distant in time could somehow move together. While in life sciences, it seems like we're always looking for certainty, or sort of falsifiable certainty. We're looking at a structure, or we're looking at something that AlphaFolds give us, and we're trying to validate it. Does the advancement in quantum physics about sort of our understanding of uncertainty influence how we're thinking about life science and doing research in life science? Thank you.
That's a tough one. Okay, we have somebody who's willing to try to answer here.
So I'm not at all a quantum physicist, but my understanding is it mostly applies to very small systems. On the other hand, this stochastic aspect also comes about when you are talking about a small number of molecules. And I think, this might have come from the others in the round. If you look at gene expression at a single cell level, you see this stochastic noise very, very, very clearly. And modeling has also shown that these stochastic fluctuations actually contribute to the robustness of a system. So this might be the closest answer to your question I can think of.
I would second that. I think stochasticity and randomness are in events. You were describing biology as being very deterministic, that we want everything on a road. And in fact, there's probability that something will happen, and then there is a lot of stochastic events, and it depends with the granularity that you look at things. That's what you were saying. If you look at few enough, everything looks stochastic. And then larger numbers, maybe that's why we need lots of numbers or lots of information. You start to see maybe probability. But I do think there's, it's not deterministic. I think that it's just a level of our understanding that makes us want the deterministic rules first.
If you think of a quantum computer and you think of how it's constructed, there has to be some physical basis for a qubit. I know of no physical basis of a qubit in biology. If you can find one, do it.
Let me come in here as well. So firstly, you don't need quantum mechanics for uncertainty. Newtonian physics is completely uncertain for three bodies. As you know so well from the Chinese book Santi, basically three suns are chaotic, uncertain, unpredictable. So it's quite clear that since we are consisting of more than two atoms, life is completely non-deterministic. Determinism, I think, was something that didn't need quantum mechanics to throw out of the way. Just the existence of these mathematical problems showed that you could never predict how three water molecules would behave for long times. That's the one side of the story. On the other hand, evolution has evolved all our systems to deal with uncertainty. So basically, we're living, we're stable, life is generally fairly stable. If you think about all the cells, I mean, we go through, I don't know how many cell divisions in a lifetime, but it's a really big number. And most of them are fine, not always. So life is incredibly stable. And certainly in the sense of evolution, you know, life has been around for 3 billion years, and it's still here. So I think evolution is a great way of selecting those aspects of the system that will give you stability. Stability is very important for evolution, because if you make a change, a mutation, and what happens is completely random, you will never evolve. A mutation has to have a small effect. If it has a huge effect every time, so the space of evolution has to be fairly smooth. But nature has done that. You know, everything in life, it's not just chance physics. It's very carefully constructed with a wonderful history. So I don't think we have to worry about this, at least not for another 3 billion years.
We have another question here.
All right, thank you so much. My name is Mi Tan. I'm currently working at a private equity firm as an investor, but not specialized in life science or medical area. This question you might get to hear a lot. It's about treatment of cancer in the future. We know that a lot of the blood disease cancers can be treated by CAR-T treatment now. It's amazing how one of my close relatives was fully cured by using this CAR-T treatment. But a lot of solid tumor or like lung cancer, liver cancer, still it's quite hard to be fully treated. So yeah, I guess this is my question. How long will it take for this hard to cure cancer to be fully treated? Yes, thank you.
There's a lot of progress. I mean, the number of people working on how to make it sensitive is in the thousands of labs. So it's actually, I'm quite close to it now. So I think we will get them one by one. There's not going to be one trick. Every tumor, even every tissue can have different types of tumors. Everyone will have a different sensitivity to the immune, or permeability to the immune system. It will be, you know, it won't be the domino effect, but we will learn bit by bit. And there will be a breakthrough, you know, with certain, you know, we'll have another insight after PD-1 and the immune checkpoint breakthrough. There'll be breakthroughs, but it still will be tumor by tumor, or tissue by tissue, I'm sure.
Yeah, I mean, the success in cancer has been enormous. My former colleague Jim Allison was responsible for the immune checkpoint therapy. Before his discovery, melanoma was a virtually certain death sentence. Now, many melanoma patients can live out a full life. So I look at it, in that case, the glass being half full, more than half full. On the other hand, neurodegenerative disease is much worse and rising in incidence. There's no treatment for Parkinson's disease. The treatment for Alzheimer's is dubious at best. People are going to be dying in increasing numbers that will soon exceed those of cancer and heart disease. That's where I think the effort should really go. Sir.
I mean, following up what Susan was saying, I think getting a cure for the major type of cancers, this is in sight, in particular as the immune system is already quite trained to deal with a cancer. But in the end, it will end up with a quite individual therapy. So the diagnosis has to be so precise that you really know what is going to work. And the question will be, is the society willing and able to invest these resources in curing a single patient? So it will turn from a medical to a social problem, I'm afraid.
Invest in prevention.
Thank you. Thank you. Many of you mentioned that. Tell us who you are. I'm sorry. I'm Tian Wei from Shanghai Tech University, and I'm a PhD student, major in neuroscience. Many of you mentioned that technology helps science a lot. Look back the past decade. Optogenetics has brought a lot of help of neuroscience research. May I ask what was your inspiration that led you into the great technology? And I want to know if any new technology and the patient one should we pay attention to the field of life science research in the next 10 years?
Did I understand you correctly? You asked about the inspiration behind optogenetics? Yes. The inspiration was the solution to a different informational problem in biology. And that is the experimental strategy that led to the breaking of the genetic code. Marshall Nirenberg and Heinrich Mattei fed the biological system simple artificial messages. RNAs that consisted of just one nucleotide, and then they observed what the system did with these artificial signals. The idea behind optogenetics is the same. We feed simple artificial signals into the nervous system, and then we see what the nervous system does with them. Whether we can somehow, like a biochemist would reconstitute a biochemical reaction, reconstitute perception, action, emotion, thought, memory, and so forth.
Other question. Okay, I'll get you next.
Thank you, professors. I'm Zhen Xinyao from Shanghai Jiaotong University. As the experts mentioned that with the development of AR models to predict the protein structures more and more precise, I wonder how much importance the experimental to determination of protein structure and what new areas can be developed, not just limited to structure prediction. That's all. Thank you.
Go ahead.
We had this discussion before, and of course AlphaFold is extremely valuable. It has helped us solve many problems much quicker, but it doesn't solve every problem. And in our area, I have to say I'm fortunate that I'm not becoming unemployed by AlphaFold, because AlphaFold does not tell us how a protein folds. and it certainly does not tell us how the cellular machinery helps a protein to fold. But as one example, we try to use AlphaFold to predict how a short segment of a polypeptide chain would fold during its translation on the ribosome, where vectorially the polypeptide chain emerges from the ribosome. And we were hoping that we could find parts of proteins that are prone to adopt alternative structures other than those that we find in the final native state of the protein. But AlphaFold gives us always the structure of the final protein because that's what it's being trained on. So we have to now try to modify, perhaps if possible, to modify the algorithm to look at just segments of a polypeptide and tell us something about the propensity of these parts to fold into either the proper structure or into alternative, because that's perhaps where the molecular chaperones would then become active in correcting these mistakes. So AlphaFold is great, but it doesn't answer every problem. It does help us, though, in doing structure-based mutagenesis. And we have another interesting example recently where we actually solved an experimental crystal structure. AlphaFold structure is very close, but it does deviate in some important parts of the structure.
If I just may add to that, the truth is that AlphaFold is developing so rapidly that what we're saying today is probably not true anymore next week. And so we really have to keep our eye on the ball and see how we can harness this predictive power to design better experiments and to really focus on the difficult structural problems. And it really opens a lot of doors to do higher throughput structural biology.
Thank you very much. Good afternoon, professors. I'm Zhou Liang, an undergraduate from East China University of Science and Technology, major in bioengineering. I feel honored to ask you a question that, as we all know, synthetic biology is an emerging field of biology. I wonder nowadays how synthetic biology influences and changes the traditional life science and medicine areas, subject area or research, and what stage has it developed so far, and where its future go? Thank you.
Can I? Yeah?
Go ahead, Danielle.
I mean, I only know from short sort of from the lab I was working, Jason Chin's work, which is amongst probably the most visionary. For instance, he has rewritten the code from 3 to 4 letters. So the ribosome, modified the ribosome, so it recognizes four bases. So that allows you to, rewrite the code of proteins. He's also figured out how to install modified amino acids through synthetic biology. So I think there is a great future in synthetic biology. It's difficult to, I think, to predict right now how far it will go, but the progress has been immense just in a few last, I would say, 5 years.
I think if the question is how does it apply to medicine, I think that takes a question by question answer, because again, you need to understand what is the medical question, what is the, the fundamental biological or biochemical problem, and then you ask, which is possible now, if we, don't follow the rules, but make new rules, can we solve the problem? So not to be, I think what synthetic biology does is allow us to go beyond the rules that we usually play by, or the cells usually play by, and create new things.
Thank you. Hello, I'm Josie Fullerton. I'm a postdoc at the University of Glasgow, and I'm here representing the Young Academy of Scotland. As a postdoc, I'm very aware of the importance of having a mentor, and I'm very lucky to have 2 or 3 of them. I was just wondering if you guys have mentors, and if anyone is looking to get a mentor, if you have any advice on how to find one that could help them. Thank you.
Yeah, I'm sort of like a key mentor for lots of women in Switzerland. So, did I have a mentor? Of course, my PhD advisor I admired greatly. Everybody I worked for, you learn something from, right? So, and no mentor actually solves your life for you. All they can do is either you can look up to them or you can say, well, that person's okay on some things, but I could do it better. So, I think the great thing is, as a mentee, which, many of you in the audience may want, find someone you admire. And maybe that you don't admire everything about this person, because there are not such people. But latch on to the best things, and then, let them listen to them, how they might have solved their own problems or guide. The other thing is networking. Mentors are good for reaching out. As you go up your life through science, you know more and more people. And networking, so that if you have a problem, knowing whom to go to is sometimes key. And I find mentors are good for that. And maybe as a mentor of many women, what I do is just put them in contact with other people, because I can't solve a lot of people's problems.
Was the question how you find a mentor, or? Yeah, I think it's difficult. One thing you can try and do is to get them to visit wherever you are. Have them come and give a seminar, talk to the students is a separate thing. I found this, for me, extremely helpful. And also, when I was leaving school as an undergraduate, I just don't know how possible this is anymore, I went to 10 different schools. I had picked people that I thought might be of interest and actually went and interviewed and talked to them for a while to find out if I could sort of live with them for 4 or 5 years. I don't know if it's not easy to do, but it certainly makes a difference.
I just want to add, if you look for a mentor, you should look for somebody who is actually willing to listen to you, because many of us, as you may have noticed, have the tendency to pontificate, and and so it's really important that you are heard, and because sometimes just articulating your problem, your conundrum to somebody else is already the solution.
Okay. Boy, we're getting more questions as the hours go on. Okay.
Good afternoon, professors. I'm Luo Chen, a freshman in the Fudan University. From my perspective, you are all successful scientists, and I want to learn from you. But now I'm confused about how to choose my direction of research, or why do you choose to make researches on the nucleosome? Thank you.
I mean, I think sometimes, it's just an accident. I really admire people, and I always tell my students, pick something you're really interested in. In my case, I landed in a place where I watched this problem being solved and realized it was very interesting. And going back to the mentor, I came across Aaron Klug. He was a fantastic scientific mentor, but at the personal level, he was extremely difficult to sort of get on with, because he was not really, he lived in his head. So that's the other thing, is to learn to adapt and make use of the people who around you in the right way. I mean, I could tell from the way Aaron walked down the corridor whether it was a good idea to talk to him or not. So if he walked in a certain way, I wouldn't bother. And I learned, in his case, the best way to communicate was to leave him a little note on his desk to say, I wanted to talk about a problem. So you have to switch them on to you. And, you know, I think it, more generally, I think lots of young people think that science is, being a scientist, like a profession. I think, and maybe it has become a profession, but certainly when I was young, it was really more of a passion, curiosity, and then it's not what you knew. You just learned what you needed to know to solve the problem. And actually, in our days, very often, we had to invent the techniques to solve the problem. You know, so you should just sit back and say, what is the most interest? If I had a chance, what would I really like to work on?
Also,
you know,
I think it's really important to try and go to, I have no way of evaluating this with each one of you, but a really good place where things are happening. And you may go to one laboratory because you think that person is really great, which is fine, and you may stay there. In my case, I did that. I wanted to work on protein folding, but somehow got hooked on protein DNA interaction. And then I knew I wanted to go to Cambridge because that's where structural biology began, and I wanted to see what that was all about. So I was offered a couple of projects, one of them being the nucleosome, and it was so fundamental in protein DNA interaction, how could I pass it up? The other was cryo-electron microscopy with Richard Henderson, which I turned down, of course. But I have to say, be careful what you wish for, because yes, it is difficult. You are expected to do the job, and as the time goes on, it becomes a huge burden, and it'll be, the success will depend on your personality, whether you can withstand that burden and thrive on it, or whether you'll collapse and decide to do something else.
I think, I don't know if you have this in China, but certainly in England we have this. Undergraduates get sort of 2 months, 3 months work in the lab during the summer vacation. That's a really good way of getting an experience of working within a research environment. So that's what I would try. If I were an undergraduate now, I would try to get summer placement in a lab.
Yeah, that's a really good idea, and don't be afraid to switch fields or approaches, and in fact do that several times in the course of your career and find out what you like best, and then you can put all of those things together and make something unique.
Okay, so thank you. Good afternoon, professors. I'm Ma Danlu. I'm from Shanghai Tech. And so I want to share this view that I read in a book recently, and I want to hear your opinions. So I can't remember the author's name, but it was a physicist, and he presented this view, a hypothesis that, in fact, everything in this university works along a predictable and reasonable way in a direction that can be explained by physics and math, right? We keep making predictions like where an atom moves or when will a person get Alzheimer's, etc. But we keep running into things that seem unpredictable, and we can't seem to find a pattern in how they move. But the hypothesis that this physicist came up with was that, in fact, for those things we refer to as unpredictable are merely things with too many variants for us to detect at once. So all the variants work in a predictable way, but we just can't detect them all at once. So that's why many things seem unpredictable to us. And so I want to ask, what do you feel about this opinion, and what do you think of possibilities and unpredictabilities?
Yeah, that's a Randy.
Again, a very interesting question. You know, I think that lots of variables are very hard for us, but some things do have lots of variables. On the other hand, you know, for every problem, in thinking about it, there's like the right number of variables. So if I move my hand, we can probably pass it into, I don't know, 20 joints or something like that. But in my hand, there are probably 10 to the 15 atoms or 10 to the 20 atoms, I don't actually know, pretty more like 10 to the 20 atoms that are all moving. So I think there's the right level of simplification, but again, I don't think that all problems have an easily explainable solution. I think many do, and it's often very difficult to find it. So I don't think there's any kind of magic where if we had all the variables, everything would be easy. Some problems are just very, very unpredictable. But again, this is more mathematics. It's like saying if something is, there's a way of ranking problems. And in biology, some things are like that. I'm not sure it's worth worrying about.
Questions, questions, questions. Okay, lots of questions. All right. We have just time for 2 more questions.
Yeah. Hello, professors. I'm Cheng Qiang from Fudan University. It's my first year to study in university. I want to ask, as a freshman, what can I do to prepare for my research in the future, such as which skills should I to cultivate? Some basic skills.
Yeah, I can answer that because I teach freshmen a lot. And,
thank you.
And my approach, my advice would be to get research experience, go into a lab so that you can really connect what you're learning in the classroom to what you're learning, what you're doing in the lab. And also the realization that the material that is in books was actually discovered by people like you. It did not fall from the sky. Things that are in the textbook are done by researchers like yourself. That is a really important realization, and I think that should really provide the impetus for you to get into a lab and try whether you like research yourself.
My own experience was simply that. In the second term of college, I actually went and worked for agricultural test station, which was at the university, in order to get some experience, to see if I liked lab. And I did pretty well there, and I could measure samples and things. And so then I went up a bit, went into an actual research lab, and every single year, every semester, I worked in a laboratory, and generally I actually got paid for it as well as a technician. And so I learned that I loved research, and I loved being in the lab. So the only way to really find out is to do it. Try and get an opportunity to go and see if you like it or not.
Super answers, super answers. One last question. What am I bid? All right, the winner is?
Good afternoon, everyone. My name is Yao Fenghou, a graduate student from Shanghai Tech University. The question I want to ask is how young students find a field of study that interests them and stick with it. Thank you.
Well, that is a bit related to the last question, I think.
We didn't understand the question.
Well, I think it was, how do you identify a field and stick with it? Is that what you asked? Yeah. So how do you find a field of interest that's so compelling that you wish to stick with it?
Read as much as you can.
Well, I would go back to the other answer. Yeah, get a broad fundamental exposure to the basics, the basics of molecular biology, cell biology, biochemistry, physics, and then try labs, work in labs, try some problems on, and if you don't like them, then move on. There's nothing inherently, it has to suit you. It's not that because the professor thinks it's important that it's necessarily important. So I agree with the statements before that nothing replaces a bit of experience in a lab, seeing how they approach the question, whether it's a tractable problem, and whether you enjoy it.
Once you're in a lab, you'll also be able to talk to the other people in other labs and see what they're doing. And with time, you will find a direction that happens to suit you, which suits your interests.
I just want to end with saying that there's a lot of different approaches to solve problems and to approach a scientific problem, bottom up, top down. There's just many different ways to skin this cat, so to speak, and you have to find an approach that works for you. And I think that's almost equally important as finding the exact problem that you want to study. It's more like, how do you approach a problem?
Thank you very much, all. I want to thank the panel for their valuable insights, diverse as they may be. They were really terrific to share with all of you, and I want to thank you, the audience, for being so robust in your questions. I will say, now reflecting on the 5 years of the WLA, I've seen quite a change in the atmosphere of the participating students. This is the first time that I've seen such a vigorous discussion coming from the audience, so you are to be congratulated as well as my colleagues, so thank you all.